In my college genetics lab this fall, we attempted molecular cloning, transferring a specific segment of DNA into a host cell to induce replication of a particular gene. But we kind of cheated. We used topo-TA cloning, a PCR-based cloning mechanism that uses topoisomerases. It's a method patented and distributed in ready-to-go kits by Thermo Fisher Scientific. And this method worked great for our lab. It was simple and fast enough to allow me to walk down from Science Hill before dark. The drawback is that this method is expensive, and most labs won't have pre-made cloning kits to use, so most people have to clone the long way. In an effort to avoid getting laughed out of a lab in the future, I took it upon myself to discover the truth about so-called traditional cloning. Step number 1, design. I figured out that you need two things to pull this off, a vector and a DNA segment. The DNA segment is what you are attempting to have replicated and expressed in your model organism, and the vector is the vessel that carries that information. It's kind of like a message in the bottle situation. Vectors are usually bacterial plasmids a few thousand base pairs in length. They also contain an origin of replication and a multiple cloning site. The MCS is a region on the plasmid that has many restriction enzyme recognition sites, and that's what we're going to use to insert our DNA of interest. Traditional cloning uses restriction endonucleases, and while they originally evolved to defend against bacteriophages, they're extremely helpful for cutting things that need to be cut. Basically, these enzymes recognize short DNA sequences and cut them to make blunt ends or sticky ends. Sticky end cloning is a more commonly used method, and it ensures that the DNA is always inserted unidirectionally. To make a DNA segment that can be placed into a vector, we need to design primers that contain the restriction site for the specific restriction enzyme we want to use. And this comes into play in step 2, amplification. The thing about cloning is we need a lot of DNA, and usually we get all that DNA using PCR, or polymerase chain reactions, invented by this guy. But that's not really important. What is important is thermal cycling. Basically, PCR relies on two temperature-dependent reactions to replicate a whole bunch of DNA. And once we have all that DNA, we can move on to step 3, digestion. Remember those restriction endonucleases? Here's where we actually use them. Our DNA and vectors now meet our chosen restriction enzymes, which make the cuts necessary for joining the two in step 5. But first, step 4, purification. With our DNA and vectors cut, there's a whole bunch of random enzymes and leftover DNA fragments that we don't really need. We run an agarose gel to separate the fragments, and we're left with what we need for step 5, ligation. To combine the gene of interest and our vector, we use an enzyme called DNA ligase. Ligase acts kind of like glue, catalyzing the formation of a phosphodiester bond between two nucleotides. It's used in a lot of DNA repair systems, and the result is a circular molecule containing the gene of interest, which can be taken in by competent cells in step number 6. Transformation. Bacteria naturally takes in some foreign DNA, but the rate at which it does this is too slow. So we make the cells more likely to take in foreign DNA using heat shock or an electric shock, and then bam, molecular clone. Oftentimes the competent cells are plated on antibiotic media to determine if the transformation was successful. The colony should grow, and we determine the transformation to be a success. Now, I don't know if I'll ever end up cloning in a lab in the future, but hopefully I know just enough not to get laughed out of the room.